Welcome back to the series of videos where I'm going through the FRQ questions, and we have reached the last one that's going to be covered on this year's abbreviated exam. And so this is unit, if you're moving sequentially through them. If you're hopping around, then I don't know. I don't know where you're at. But if you're going sequentially, this is the second FRQ for Unit 6, which is gene expression and regulation. Again, that is stuff that we've been doing in class very recently. So it corresponds with 16, 17, and 18, with a little bit of chapter 20 in there. Biotechnology, can kind of be inserted as a context for lots of different questions. Okay, so let's take a look at question 2 here. So DNA replication in eukaryotes and prokaryotes starts when a complex of proteins binds to an origin of replication and separates the two strands of the double helix to form a replication bubble with replication forks at each end. In order for replication to proceed, the enzyme topoisomerase binds to the DNA immediately in front of the advancing replication forks. So this should look pretty familiar because you guys did a DNA model yourselves where you modeled a replication fork, and then we put them together with the group next to you, and so you had a replication bubble like this. And then I attempted to hang them all outside in the hall, but they left the heat on and it dried out all the masking tape, and they were all on the floor Tuesday, and I cried. But that was my intent. But you guys saw at least how they lined up with your neighbors. So you should at least got to see one of these replication bubbles in action. So let's see what they want to have you do. So describe how the structure of a prokaryotic chromosome differs from the structure of a eukaryotic chromosome. Okay. So generally, as you guys have seen when we used plasmids, right, is that bacterial DNA is going to be generally circular and just have one point of replication. Like it's going to have one sort of origin of replication, as they say, and it's just going to, separate. The enzymes are going to zip around, meet kind of over here, and then you're going to have two new circular pieces of DNA. Whereas eukaryotic is going to be linear and very long, right? Oops. So, obviously, tools for drawing on this aren't great. Mr. Wise tells me I got to buy one of these tablets, but I haven't done it yet. So if this is a replication bubble, right, it just, this is what you're seeing here. It means that in a eukaryotic chromosome, there's a lot of these because they're all sort of happening at once, right? They're all sort of happening at once. So you'll have, one here, and then you might have one down here, right? Over here somewhere. And, this one would be going this way, and then this one would be going that way. And then these two, right, would actually meet. So, yeah, I think the most important thing is that, prokaryotes, like your archaea and your bacteria, circular DNA, and then sort of one origin of replication. Whereas eukaryotes are going to have linear chromosomes. I guess this should really be chromosome. Linear chromosomes, many origins of replication. Because it would take too long if there was just one trying to do the whole thing. Because, you guys have, there's that whole 6 feet of DNA in every cell that you gotta, that you have to replicate every time your cell divides. So, like, in molecular terms, that's a ridiculous distance. So you got to have many origins of replication in order to get the job done in a decent amount of time. Okay, so explain why DNA replication cannot proceed to the regions of the chromosome labeled as 1 in figure 1b unless topoisomerase binds ahead of each advancing replication fork in region 2. Okay, well, when you guys made your models, you guys showed this, that your main, because in order to get this open for replication, you need DNA helicase, because as you guys all know, DNA is in a double helix, and helicase has to go through and, unzip your genes. So helicase has to go through and unwind the double helix and actually break those hydrogen bonds that are holding these two strands together. And if you think about that, if helicase did that, it could damage the DNA up ahead, because if you've got helicase unwinding this DNA, you may get the helix is going to get twisted up ahead of that. So what topoisomerase does is it moves ahead of the helicase and sort of stabilizes the double helix that has not yet been unwound. So topoisomerase, they call it relieving torque, technically. Topoisomerase has to move ahead of helicase to relieve the stress. The stress caused by the unwinding of the helix. Cool. So let's just double check that real fast. So yeah, prokaryotic chromosome circular, eukaryotic chromosome is linear. I guess they didn't care about origins of replications at all. Thanks for nothing, College Board. So the response indicates that as the DNA helix unwinds, it becomes increasingly tightly coiled or supercoiled ahead of each replication fork, right? And then topoisomerase relieves the supercoiling, so it stabilizes the unwinding of the double helix. So pretty much what we had said. I'm kidding, College Board. We love you. Use the template of a replication fork to draw arrows that represent both continuous and discontinuous DNA synthesis. Draw one long arrow to show the continuous DNA synthesis, and 3 arrows to show discontinuous DNA synthesis. The arrows should point in the direction of nucleotide addition, and the 3 arrows showing discontinuous synthesis should be numbered 1, 2, and 3 in the order of fragment synthesis. Well, great. This is exactly like the model that you made in class. It's nicer looking, possibly, but content-wise, you've done this. So this is actually you should have tried this already, and you shouldn't even be watching this video because you should have done so well. Okay, so as you guys know, DNA polymerase 3 DNA polymerase 3 is going to be our workhorse, and it's going to be the one that's laying down the nucleotides. And as you also know, the new strand of DNA is going to be anti-parallel to the old one. So if this is the 3' end of the old strand, it is going to be the 5' end of the new one. And so again, in a replication fork, you have, and as you guys did in your model, you had helicase here, moving this way, right? And then since nucleotides, DNA polymerase 3 can only add nucleotides in the 5' to the 3' direction, it is going to synthesize DNA continuously. And this is, of course, what you guys had labeled on your diagram as the leading strand. And then, continuous synthesis, right? Because the DNA polymerase is there has to be a primer here, obviously, but it can attach to the 3' end of the primer. and then just 5 prime to 3 prime, 5 prime to 3 prime, 5 prime to 3 prime, and just zip along after the helicase. Easy, right? Now, the other one is a little bit more problematic because the 5 prime, it still has the same limitations as this, in that it can only go from the 5 prime to the 3 prime direction. And so same problem, same thing here, but the difference is here, since the 5 prime to 3 prime direction is that way, DNA polymerase can move that way. But up top, it's more complicated because it can move, it can only move this way. But the helicase is still moving this way. So the hard part of your model, if you remember, was thinking through time. In like, you know, what had already happened, what happened the most recently. And that is actually the very thing that this question is asking you to do, almost exactly. So as you guys put in your model earlier, it would be this way, and this one would actually happen first. So this is our 1, right? Then, because as you recall, you know, primase, DNA polymerase 3, right, would come through and go this way, and then they would loop back and then synthesize another short piece, which are, you had labeled as Okazaki fragments, aka Yamasaki fragments. And then it would do so again. And so you would have the most recent one closest to the replication fork, because the overall direction of synthesis is still this way. But this is going to be our lagging strand, which is going to be discontinuous synthesis, because it has to be synthesized in pieces. It looks like a lot of vowels, but I think that's right. Discontinuous synthesis. Okay. Cool. I feel like that's pretty thorough for part C. Let's look at part D, and then we'll be done with this whole thing. Okay, referring to figure 1b, explain why any newly synthesized strand of DNA is the result of both continuous and discontinuous DNA synthesis. Well, it has to do with the relative orientation of the replication fork. Because you got to remember that looking at this, right, it just depends on which way the helicase is going, right? Because it's not going to change. Like this whole thing is going to be 3 prime over here to 5 prime over here, right? This strand is going to be, 5 prime to 3 prime. So if the helicase is moving the other way, then up here, right, the one that can follow the helicase is the one that I can actually move left, right? So on this side of the replication fork, this would just be able to synthesize continuously in this direction, where it would be this one that would then have to loop back, right? So it's all relative, really. So I guess the best way to say that is just going to be like, continuous or discontinuous replication is dependent on the direction of the helicase. And will be different depending on which half of the replication fork in each half, I guess, in each half of the replication fork, but replication bubble. So let's actually see what they were looking for there, because I'm not entirely sure how they would like you to explain that. I think I did an okay job. All right. So the response indicates the presence of one long arrow drawn on the lower strand of the template and pointing to the right. Done. Continuous DNA synthesis. The response also indicates the presence of 3 arrows drawn along the upstrand, pointing to the left. The discontinuous DNA sequence synthesis occurs along this template strand. From left to right, the arrows should be numbered 1, 2, and then 3 to kind of indicate the overall direction of replication. The response indicates that because DNA synthesis only occurs in a 5' to 3' direction, synthesis of a DNA strand in a replication bubble is continuous at one replication fork and discontinuous at the other replication fork. Yeah, I suppose I should have mentioned something about 5' to 3', just because it seems like a mantra of this particular part of biology. But it's what we said is that, since the DNA polymerase can only move in that 5' to 3' direction, it just depends on whether it can move with the helicase or in the opposite direction of the helicase. Okay, well, we have made it to the end of this video series. I hope it was helpful. So our school officially starts up on April 7th, which is going to be next Tuesday. So I will certainly be communicating with you guys between now and then. But hopefully this will tide you over, so to speak, if you're looking for some way to get ready for the AP exam before we start kind of officially going back to school. Can't wait to see you guys, or at least via Google Meeting or however we decide to do it. So I'll be in contact with you soon. Thanks.